Welcome to GE Fun Facts Online. Today we are not looking at the stars, nor are we looking at the latest silicon chips. We are descending into the shadowy recesses of the world's tropical oceans to witness a silent war. It is a war that has been raging for millions of years, fought not with claws or teeth, but with some of the most sophisticated molecular cocktails known to science. We are talking about the future of pain management, a future that might not come from a laboratory synthesis, but from the venom ducts of a tiny, neglected marine predator. This is the story of the Turridrupa snail, and how the bioprospecting of its venom might just solve the opioid crisis. For decades, pharmaceutical researchers have looked to the rainforests for the next generation of medicines. We have scoured the canopy for alkaloids and the soil for antibiotics. But a paradigm shift is currently underway. The medical community is desperately scrambling for alternatives to opioids. We all know the statistics. We know the devastating risks of addiction and respiratory depression that come with morphine, oxycodone, and fentanyl. The holy grail of pharmacology right now is a non-addictive analgesic, a painkiller that turns off suffering without turning off the person. And the blueprint for that drug may lie hidden in the complex biology of marine snails. Now, if you follow marine biology or pharmacology, you have likely heard of the cone snail or conus. The cone snail is the celebrity of this world. It is large, it has a beautiful shell, and it produces a venom so potent it can kill a human. Science has already successfully mined the cone snail to produce ziconotide, a drug approved by the FDA for chronic pain. But focusing only on the cone snail is like walking into a library and only reading the books on the bestseller shelf. The cone snail represents only a tiny fraction of the ocean's venomous diversity. Emerging from the shadows is a less understood, yet potentially more chemically diverse group, the turrids, and specifically the genus Turridrupa. To really understand why this is such a massive breakthrough, we have to look at the family tree. The superfamily Conoidea includes three main groups. You have the cone snails, the auger snails, and the turrids. As I mentioned, cone snails are the heavy artillery. They are the tanks. They blast their prey with massive doses of paralytics. Because they are large and easy to collect, science picked the low-hanging fruit. We have cataloged thousands of conotoxins from them. But the turrids? They are the dark matter of this universe. There are over 10,000 species of turrids, that is 10 times the number of cone snail species. Yet, amazingly, less than 1% of their venom peptides have been sequenced. They were ignored for a century because they are tiny, often less than 5 millimeters long, and they live in deep, inaccessible waters or cryptic coral reef environments. In the past, you needed to milk thousands of snails to get a sample. Today, however, we have the revolution of venomics. Using high-sensitivity mass spectrometry and transcriptomics, we no longer need liters of venom. We can sequence the DNA and RNA from a single venom duct of a single tiny snail to predict the entire library of toxins it is capable of producing. This technology has blown the door open on Turridrupa bioprospecting. Before we dive into the chemicals, we need to address a bit of detective work regarding the names. Taxonomy in marine biology is always shifting as our genetic tools get better. The genus Turridrupa has undergone significant scientific revision recently. Through modern molecular analysis, several species that were historically classified under Turridrupa have been reclassified into the genus Crassospira. This matters because if you go looking for the research papers on these new painkillers, you might not find them under Turridrupa. Much of the groundbreaking research is technically published under Crassospira. For example, the star molecule we will discuss later, CCE9A, was isolated from Crassospira cerathena, which used to be known as Turridrupa cerathena. For the sake of clarity today, we will discuss them in the context of turrid venoms generally, but keep in mind that science is essentially rebranding these creatures as we learn more about their genetics. So what makes their venom so special? If cone snails are the heavy artillery, turrids are the snipers. Their venom composition offers a completely new chemical palette for drug designers. This comes down to structural innovation. Cone snail venom is famous for being rigid. The molecules are held together by what we call disulfide bridges. Imagine a protein like a piece of string. A disulfide bridge is like putting a staple through two parts of the string to hold it in a loop. Conotoxins have many of these staples, making them very stable and very specific. But turritoxins, the peptides from our turrid snails, often break this rule. Recent studies from the University of the Philippines and the University of Utah have revealed that many turrid peptides lack these extensive disulfide frameworks. Instead, they rely on other structural motifs, like alpha helices, to maintain their shape. This might sound like obscure chemistry, but for drug manufacturing, it is a game changer. Disulfide bonds are difficult and expensive to reproduce in a lab. If you are trying to manufacture a drug at scale, you want something simple. Peptides that don't rely on complex bridging patterns are often cheaper and easier to synthesize. Furthermore, a different shape means they bind to receptors in the human body differently. If a conotoxin blocks a pain receptor by plugging the hole like a cork, a turritoxin might work by jamming the hinge. It offers a new way to shut down pain signals that the body hasn't developed a tolerance to. We also see a chemical anomaly in these snails called the methionine mystery. Turrid venoms have a surprisingly high prevalence of methionine, tyrosine, and arginine residues. In typical biology, methionine is often just a start signal or structural filler. In turritoxins, it appears to play a functional role in stabilizing the toxin's active coiled coil structure. This represents a novel structural class of neurotoxins. In the world of pharmacology, the word novel translates directly to opportunity. It means these molecules might evade the immune system or cross biological barriers more effectively than what we have seen before. Let's move from the theoretical to the concrete. The potential of this bioprospecting became reality with the isolation of a specific peptide named cresapeptide, CCE9A. This is a 29 amino acid peptide. It was purified from the chaotic mixture of hundreds of toxins found in the crude venom so that researchers could test its specific biological effects. And the results were fascinating. When they introduced this peptide to mouse models, it didn't just paralyze the mice. If it had simply paralyzed them, it would be a boring, lethal toxin. Instead, it caused a phenotypic shift from lethargy to hyperactivity, depending on the age of the mouse and the dosage. Now, hearing hyperactivity, you might think, wait, I want a painkiller, not a stimulant. But in neuropharmacology, this is a gold standard signal. It proves the peptide is psychoactive. It proves that the molecule is successfully crossing into the nervous system and modulating the firing rates of neurons. It isn't just destroying tissue, it is flipping switches in the brain. The mechanism of action here is incredibly precise. The specific target of CCE9A and related turritoxins appears to be the nicotinic acetylcholine receptor, or NACHR. These receptors are the primary communication gates between nerves and muscles, and crucially, they regulate pain pathways in the central nervous system. Think about how opioids work. Opioids generally work by dampening the signal after it has already arrived at the processing center. Turrid peptides, by targeting these nicotinic receptors, essentially stop the pain message from ever being written. It is similar to how nicotine can have mild analgesic effects, but these snail peptides are thousands of times more potent and specific. They target only the receptor subtypes involved in pain sensation, without affecting the ones that control your heart rate or your breathing. That specificity is the key to avoiding the side effects that plague current pain medication. This brings us to the medical logic of the future. The leap from a snail killing a marine worm to a human taking a pill for back pain relies on homology, the shared biology between species. The ion channels that a worm uses to wiggle are remarkably similar to the ion channels humans use to signal chronic neuropathic pain. The search for non-opioid analgesics is largely a search for voltage-gated ion channel blockers. The drug ziconotide proved this works by blocking N-type calcium channels. But ziconotide has a major flaw. It cannot cross the blood-brain barrier effectively. It has to be injected directly into the spinal fluid. That is a painful, invasive procedure. Turrid peptides offer a second chance. Because they are structurally distinct, often smaller or helical, there is hope that they can be engineered into peptidomimetics. These are drugs that mimic the peptide, but can be taken orally. Specifically, researchers are looking at the alpha-9 alpha-10 subtype of nicotinic receptors. This specific receptor is a prime target for breast cancer pain and chemotherapy-induced neuropathy. If a turridrupa peptide can selectively block this receptor without blocking the ALPHA7 receptor, which is needed for cognitive function, we could have a painkiller that doesn't cause drowsiness, addiction, or brain fog. Furthermore, recent papers have highlighted that turrid venoms rely on what they call synergistic cabals. This means the snail doesn't just shoot one bullet, it fires a team. Toxin A might open a hole in the cell membrane. Toxin B slips through that hole to block the pain receptor. Toxin C prevents the enzyme that would normally break down toxin B. Bioprospecting is now looking at using these helper peptides to improve drug delivery. Even if the analgesic peptide itself is hard to deliver to a human nerve, the Turridrupa snail might have already evolved a delivery vehicle peptide that we can borrow. Of course, there are challenges. We face the glass ceiling of peptide drug development. Peptides are fragile. If you swallow a peptide, your stomach acid treats it like a piece of steak and digests it. This is why insulin must be injected. The solution researchers are developing is cyclization. Chemists are learning to tie the ends of these linear snail peptides, turning them into loops that enzymes cannot easily chew up. The unique, non-disulfide structure of some turritoxins makes them excellent candidates for this chemical hardening. And because turrids are too small to farm, we cannot harvest the venom naturally, we must synthesize it. Fortunately, peptides like CCE9 fall into the sweet spot for solid-phase peptide synthesis. They are small enough to be made cheaply in a machine, but large enough to be highly specific. Looking at the future outlook, The next decade of marine pharmacology is going to be exciting. Between 2025 and 2027, we can expect the publication of the first comprehensive Turridrupa and Crassospira venoms, libraries mapping thousands of peptide sequences. Preclinical trials will likely focus on neuropathic pain, phantom limb pain, and diabetic neuropathy. These are conditions where opioids are notoriously ineffective, but where channel blockers shine. And finally, there is the conservation argument. This research provides a powerful economic argument for conserving coral reefs. A single extinct species of Turridrupa could mean the loss of a cure for cancer pain. The biodiversity of the Philippines and Indo-Pacific, where these snails thrive, is now viewed not just as an ecological treasure, but as a pharmaceutical vault. In conclusion, Venom Bioprospecting is rewriting the rulebook of pain management. For too long, we have relied on the poppy flower to numb human suffering, at great cost to society. The Turridrupa snail offers a different path. Its venom, honed by millions of years of evolution to subdue swift prey, contains molecular keys that fit the locks of our own nervous system. By studying peptides like CCE9A, scientists are uncovering a new class of analgesics, drugs that are potent, non-addictive, and structurally novel. The Turridrupa snail may be small, and it may have been neglected by science for a century, but its time has come. In the war against chronic pain, this tiny marine predator is emerging as one of humanity's most promising allies. The future of analgesia is not synthetic, it is oceanic. Thanks for listening to G Fun Facts Online.